When the EEB seminar went virtual in the spring, I think, we acknowledged that maybe there was one positive side to this, and that was the potential to cast our net much more broadly than our modest budget normally allows us, bringing in speakers from more than a few hundred kilometers away. And so I think today's guest really underscores that potential, coming to us all the way from the IISER in Mohali, India. Manas, I don't know you very well personally. We've only met over Zoom a little bit, but I do know a whole lot about your academic pedigree, as well as some other things from other sources that I might mention, as you cringe. But let me just tell you, in that academic pedigree, I would be sort of like the great uncle. And that's because my connection begins way back in my PhD in the Rose Muller lab at UC Irvine, where I became a lifelong friend and colleague of Amitabh Joshi, one of the postdocs in that lab. Together, we learned about evolutionary genetics, experimental evolution, sexual conflict, and of course, lots and lots about fruit flies. When Amitabh set up shop in the prestigious JNCASR in Bangalore, India, he started training some really outstanding students. And when it came time for me to set up shop here at Queen's, it was Amitabh's student, NG Prasad, who, like our speaker today, is coming to us from far and away in Mohali. He came to my attention as a prospective postdoc. He flourished here. He really helped set the agenda for my lab in the early days. And he's gone on to become an illustrious researcher, editor, even a dean at Mohali at the IISER. So Manas is coming to us from Prasad's group, where, to continue that pedigree analogy, he was like an older brother and mentor to Harsha, PhD student who's here working with Troy Day and myself. Brother too to Imran Syed, a PhD student, pretend PhD student who's supposed to be here, but for this wretched pandemic holding him up. So we've got a big cross flow going on between the lab groups. I would love to take my sabbatical next year, hop on a plane and come and visit and perhaps meet in person and meet all of you in person, because it's way, way overdue. And I feel like our labs are such interestingly parallel enterprises. So with no further ado, I'll turn you over to Manas Arun. He is coming to you now from late at night in the foothills of the Himalayas. And I'm very, very pleased to have him this evening in Mohali. Take it away, Manas. Thank you.
Thank you, Professor Chippindale, for that kind introduction. And I would also like to thank Professor Chippindale, Harsha, and the organizers for hosting me tonight or today. I'm really happy and I feel really privileged and lucky to be able to share my work with you. So let me begin my talk with one of the simplest possible models of natural selection. Imagine you have a large randomly mating population, and let's say there is a locus A, and there are two different alleles present in the population, A1 and A2. And let's say W1 star and W2 star are the marginal fitnesses of A1 and A2 respectively. Then population genetics 101 tells us that the change in the frequency of A2 over one generation should be given by this formula. A bit of standard calculations then take us from this equation to this one, which is called the Wright's equation. Wright's equation expresses the change in allelic frequency as a function of a partial derivative of the average population fitness relative to the allelic frequency. What this equation tells us is that populations are expected to evolve in such a way that the average population fitness should increase. And more importantly, at evolutionary equilibria, where delta Q is supposed to be 0, the average population fitness should be maximized. However, real life is rarely as straightforward, and there can be a million different ways in which Wright's equation can go wrong. And one of the ways in which this can happen is frequency-dependent selection, where fitnesses of various genotypes are not constant, but are functions of allelic frequencies themselves. When that happens, the calculation that takes you from this equation to the Wright's equation changes slightly so that you get a few additional terms, which means that at evolutionary equilibria, the average population fitness is no longer expected to be maximized. A famous example of frequency-dependent selection is Batesian mimicry in butterflies like common mormon. Common hormone females can be of three distinct kinds. You can either have the stickiest morph, which mimics a butterfly called common rose, which is toxic to birds. The Romulus morph mimics a different toxic butterfly called the crimson rose, and there is a non-mimetic Cyrus morph. The important bit here is that the fitnesses of each of these different variants are not constant, but they're crucially dependent. They are crucially dependent on the relative abundance in the population. For my PhD, however, I study a slightly different set of phenomena, which are also supposed to lead to a situation where the average population fitness rather is not expected to be maximized. And these phenomena are broadly called sexual conflict. While sexual conflict as a concept has been around for probably as long as evolutionary biology, it was first mathematically formulated by Jeff Parker in 1979, who defined it as a conflict between the evolutionary interests of the individuals of the two sexes. Broadly, sexual conflict can be any situation which exhibits a negative covariance for fitness between the two sexes. Conceptually, sexual conflict can be of one of two distinct kinds. You could either have inter-locus sexual conflict or you could have intra-locus sexual conflict. Inter-locus sexual conflict is usually modeled as a conflict over mating rates, with male fitness pretty much increasing indefinitely with mating rates, while females having an intermediate optimum. Mating rates themselves are modeled as a function of a set of traits that are limited to males, which are usually called persistence traits, and a different set of traits that are limited to females, usually called resistance traits. And the underlying loci that code for persistence in males are thought to be expressed only in males, whereas the underlying loci that code for resistance are thought to be sex limited in their expression in the sense that they're limited only to females. Therefore, interlocus sexual conflict is a conflict between one set of loci that are limited in their expression to males and a different set of loci that are limited in their expression to females. Sexually antagonistic coevolution, a process where one sex evolves traits that leads to an increase in the average fitness of that sex, but leads to a reduction in the average fitness of the other sex, which results in the counter adaptation in the other sex, is a hallmark of interlocus sexual conflict. While interlocus sexual conflict has been documented in a wide range of taxa, water striders are kind of the poster taxon for interlocus sexual conflict, with intense conflict over mating rates. In many species of water striders, there is what is called a pre-mating struggle, in which males attempt to copulate with females, while females physically attempt to resist male copulation attempts. And this conflict has resulted in the evolution of a very fascinating set of traits, both behavioral as well as morphological. For example, in one species of water striders, the male antenna has been modified in such a way that there are certain specialized structures on the male antenna, like the pad or the hook or the spike, which fit exactly at specific points on the female's head, which enables the male to grasp females efficiently during copulation. While interlocus sexual conflict is primarily defined over mating rates, it is fairly straightforward to extend it to other aspects of interactions between males and females. For example, in haplodiploid organisms, you can have interlocus sexual conflict over sex allocation. Haplodiploid males benefit if the females they mate with produce an extremely female biased sex ratio, because remember, haplodiploid males do not contribute anything through the male progeny of their mates. On the other hand, the optimum sex ratio for females is determined by the local mate competition experienced by their offspring, and therefore potentially would have a situation where the optimum sex ratio for males is vastly different from the optimum sex ratio for females. Interestingly, interlocus sexual conflict is also not limited to organisms with separate sexes, with many studies having documented interlocus sexual conflict in hermaphroditic organisms. For example, in the freshwater snail Limnia stagnalis, snails that were exposed to components of male ejaculates, for example, sperm or accessory gland proteins, or a combination of the two, exhibited significantly higher levels of typical male avoidance behaviors relative to control snails that were not exposed to these ejaculate components. Even plants have been shown to be involved in interlocus sexual conflict. For example, in Collinsia heterophylla, there appears to be a conflict over the time at which the stigma should become receptive to pollen, with delayed stigma receptivity favoring female function of the plants, while early stigma receptivity being favorable for the pollen that get deposited on the stigma. Intra-locus sexual conflict, on the other hand, is a conflict over traits that are shared between the two sexes, but experience vastly different fitness optima in the two sexes. At the level of a locus, intra-locus sexual conflict happens when the allele that is favored in males is different from the allele that is favored in females. And therefore, if the strengths of selection in males and females are sufficiently balanced, you could have a situation where the population exhibits a polymorphic equilibrium such that both kinds of alleles are present in the population and neither sex can reach their adaptive optimum. Intersexual genetic correlation for fitness, or RMFGW, is thought to be an indicator of intralocus sexual conflict, with negative values of RMF being indicative of strong intralocus sexual conflict. However, some people have argued against this. Intralocus sexual conflict is typically resolved by the evolution of sex-specific expression, eventually leading to the evolution of sexual dimorphism. Intralocus conflict has been reported in a wide variety of taxa, just like interlocus sexual conflict. For example, there appears to be a strong negative correlation for fitness between fathers and their daughters in a wild population of red deer on the Isle of Room. A large number of different traits too have been documented to be involved in intralocus sexual conflict. For example, in a laboratory population of Drosophila melanogaster, there is a strong negative genetic correlation between males and females for tolerance to a pathogenic bacteria. Even plants have been shown to be involved in intralocus sexual conflict. For example, in Silene latifolia, larger leaves are favored in female plants, but the opposite is favorable to male plants. In Atlantic salmon, there appears to be intralocus sexual conflict for the age of reproductive maturation, which is essentially the time an individual spends at sea before returning to their natal streams to reproduce, with the optimum age of reproductive maturation being longer for females than for males. There was an interesting study that actually was able to identify a locus that mediated this intralocus sexual conflict over the age of reproductive maturation. And more interestingly, this study was able to show that this locus exhibited what are called sex-specific dominance reversals, in which, for example, the allele that was associated with longer ages of reproductive maturation, in other words, the female beneficial allele, was dominant in females, but it was recessive in males. To summarize, we can have two distinct kinds of sexual conflicts. You can either have interlocus sexual conflict, which involves sex-limited traits. Persistence traits are thought to be limited to males, whereas resistance traits are thought to be limited to females. On the other hand, you could have intralocus sexual conflict, which by definition involves traits that are shared between the two sexes. And therefore, at least in these formalisms, interlocus and intralocus sexual conflicts appear to be mutually exclusive. However, again, biology is rarely that straightforward, and there is all kinds of crosstalk going on between phenomena that at first glance appear to be mutually exclusive. So a big part of my PhD involves looking at the interactions between interlocus sexual conflict and intralocus sexual conflict. Most arguments in favor of an interaction between the two revolve around the idea that traits involved in interlocus sexual conflict may not necessarily be fully sex limited in their expression, leading to an interaction between the two. And there are several ways in which this interaction could pan out. For example, there could be genetic correlations between traits involved in intralocus sexual conflict and interlocus sexual conflict, which would mean that if the nature of selection on interlocus sexual conflict changed, that would have cascading effects on the intralocus sexual conflict in the population. Second, you could have negative pleiotropic consequences of resistance and persistence traits in the opposite sex. For example, females could evolve greater resistance to male courtship by evolution of modifications to their sensory apparatus. But tinkering with the sensory apparatus could potentially have negative consequences to the fitness of males in other contexts. And lastly, mechanisms that resolve intra-locus sexual conflict could trigger inter-locus sexual conflict in the population. Remember, intra-locus sexual conflict gets resolved when there is evolution of sexual dimorphism, but the evolution of sexual dimorphism could involve exaggeration of male traits to such an extent that they now become harmful to the females they encounter. So we investigated, or at least attempted to investigate, the interaction between inter-locus and intra-locus sexual conflict in the fruit fly, Drosophila melanogaster. Drosophila melanogaster is a cool system to investigate this interaction because it has been at the forefront of studies investigating both kinds of conflicts. For example, Chippendale and Rice reported in a laboratory population of Drosophila melanogaster that there was a significant positive genetic correlation between male fitness and female fitness at the juvenile stage, where sex roles are not really distinct, but the same correlation turned out to be negative at the adult stage, where sex roles are divergent. Work done in our lab suggests that there is intense inter-locus sexual conflict for Drosophila melanogaster. For example, a former graduate student in our lab, Bodhi, evolved populations of Drosophila at either male bias sex ratios, where inter-locus sexual conflict is intense, or at female bias sex ratios, where inter-locus sexual conflict is relatively weak. And after several dozens of generations of evolution, he found that males from the male bias populations evolved higher courtship frequencies, they became more active, they evolved greater sperm competitive abilities, but as a consequence, they also appeared to harm the females they encountered more. On the other hand, females from the male bias sex ratio populations evolved greater resistance to male harm. One other reason that makes Drosophila melanogaster a cool system to investigate the interaction between inter and intra-locus sexual conflicts is that there is a ready-made suite of traits that is potentially linked to both kinds of conflicts. For example, it is fairly straightforward to define and measure mate harm in laboratory populations of Drosophila. For our study, we defined mate harm as the relative decline in female fecundity between female bias sex ratio and male bias sex ratio. Additionally, you can measure a large number of proxies for male persistence and female resistance. For example, you could put a virgin male and a virgin female together in a vial and sit and watch how long they take before they start mating, which is what we call mating latency. When virgin females and virgin males are paired, they usually start mating in a few minutes. Notice that longer mating latencies are associated with higher resistance in females, but shorter mating latencies are associated with higher persistence in males. Alternatively, you could put a male with an already mated female and again sit and watch how long they take to mate. In Drosophila melanogaster, mated females are very reluctant to mate, and therefore this usually takes several hours. We call this remating latency. And lastly, you can also put a certain number of males and females together at appropriate sex ratios, and either using snapshot sampling or if you're up to it, through continuous observations, try and estimate the mating rates in those vials. There is also a nice collection of traits that are shared between the two sexes and are sexually dimorphic. For example, you could measure body size, or you could measure development time, or you could measure locomotory activity. An important bit of information here, locomotory activity has already been shown to be involved in intra-locus sexual conflict. Long and Rice were able to show that locomotory activity is a shared trait in a very technical sense of the word, in the sense that they found a strong positive genetic correlation between male activity and female activity. But the nature of selection on males and females for activity was extremely different. More active males had higher fitness, but less active females had a higher fitness. So we investigated the interaction between inter-locus and intra-locus sexual conflicts in a large outbred laboratory-adapted population of Drosophila melanogaster called LH. And we used a cool experimental technique called hemiclonal analysis, which was developed by Bill Rice in 1996. Hemiclonal analysis basically uses a certain kind of special flies called clone generator flies. Clone generator females have a compound X chromosome and a translocation between the two major Drosophila melanogaster autosomes, which are chromosome number 2 and chromosome number 3. This system, coupled with the fact that Drosophila melanogaster males do not undergo molecular recombination, results in a situation where you can pass entire haploid genomes from father to son without them being destroyed by recombination, with the minor exception of the dot chromosome or the chromosome number 4. So basically, using hemiclonal analysis, you can sample a certain number of hemigenomes from your source population, and then you can express these hemigenomes in males and females carrying a complementary haploid genome, which is randomly sampled from the same base population. And subsequently, you can measure for each hemigenome in males and females, whichever traits you want to measure. So using this system, we first asked the question whether experimentally changing the intensity of inter-locus sexual conflict influences the signal of intra-locus sexual conflict in the population. To address this question, we sampled a panel of 39 hemigenomes from the LH population and measured the contribution of each hemigenome to male fitness and female fitness in 3 different environments. Male bias sex ratio, where inter-locus sexual conflict is intense, female bias sex ratio, where interlocus sexual conflict is weak, and equal sex ratio, where interlocus sexual conflict is kind of intermediate. Subsequently, we measured 2 different parameters roughly corresponding to the strength of intralocus sexual conflict in the population. First, we measured the intersexual genetic correlation for fitness, or RMFGW, and we also measured the proportion of sexually antagonistic variation for fitness, or antagonism index. Antagonism index is a useful indicator of intralocus sexual conflict because it partitions fitness variation along 2 different axes, the sexually antagonistic axis and the sexually concordant axis. In this cartoon, you have male fitness on the y-axis, female fitness on the x-axis, and the diagonal purple line is the axis of sexually antagonistic fitness variation. Because notice that if you're moving along this axis towards the top left corner, male fitness increases, but female fitness decreases. On the other hand, if you are moving in the opposite direction towards the bottom right corner, female fitness increases, but male fitness decreases. On the other hand, the orange diagonal line is the axis of sexually concordant fitness variation, because if you're moving to the top right corner, then the fitness of both sexes is increasing. But if you're moving to the bottom left corner, the fitness of both sexes is decreasing. And so basically, once you have your fitness data for each hemigenome line, you can calculate the variance along the sexually antagonistic axis. You can calculate the variance along the sexually concordant axis, and then estimate the proportion of variance that is along the sexually antagonistic axis, which is basically what the antagonism index tells you. This is what our actual results look like. So again, on the y-axis, you have standardized male fitness. On the x-axis, you have standardized female fitness. And the dashed line indicates the axis of sexually antagonistic fitness variation. Blue represents female bias sex ratio. Red represents male bias sex ratio. And yellow represents equal sex ratio. The first thing to notice in this graph is that irrespective of sex ratio, the intersexual genetic correlations for fitness were positive. To be specific, at equal and male bias sex ratios, RMFs were significantly greater than 0. For the female bias sex ratio, RMF was positive, but it was not significantly different from 0. We were very surprised to see this result because remember, Chippendale and Rice had reported a strong negative RMF for fitness in a similar laboratory population of Drosophila melanogaster. And in fact, there have been several subsequent studies which have either reported negative RMFs or RMFs that were not significantly different from 0. The other insight from this experiment was that at higher intensities of interlocus sexual conflict, we found a weaker signal of intralocus sexual conflict. For example, if you're talking about RMFs, then the RMFs in the male biased and equal sex ratio were higher than in the female biased sex ratio. However, this difference was not statistically significant. Similarly, if you're talking about antagonism index, then the antagonism index in the equal and male bias sex ratio was lower than in the female bias sex ratio, but of course, this difference was not statistically significant. To summarize, we found positive RMFs for adult fitness at each sex ratio. And as I said before, this is a fairly surprising result because earlier similar studies had reported something different. We also found that increasing the intensity of interlocus sexual conflict in the population resulted in a slight amelioration of the intensity of intralocus sexual conflict in the population, which is also a slightly surprising result, especially given the fact that if resistance and persistence traits have negative consequences to the opposite sex, then you would expect a strengthening of intralocus sexual conflict upon strengthening the interlocus sexual conflict in the population. We were wondering why this might be happening, and we were kind of able to come up with a few testable hypotheses. First, it is possible that resistance traits could be positively genetically correlated with persistence traits. Imagine a situation where the most resistant females preferentially mate with the most persistent males, and therefore you would start developing positive genetic correlations between persistence and resistance. Now, if you artificially increase the intensity of interlocus sexual conflict in the population, which means by definition, you're going to increase the strength of selection on resistance and persistence, then all else being equal, the proportion of sexually concordant fitness variation should increase, therefore yielding a weaker signal of intralocus sexual conflict. Second, for whatever reason, instead of negative consequences, you could have positive consequences of resistance and persistence traits to the fitness of the opposite sex. And lastly, as you change the intensity, as you increase the intensity of interlocus sexual conflict while moving from female bias sex ratio to male bias sex ratio, it is possible that selection gradients on traits that are shared between the two sexes change in such a manner that selection on these traits switches from being sexually antagonistic in the female bias sex ratio to being sexually concordant in the male bias sex ratio. To kind of investigate these hypotheses, we measured a suite of potentially sexual conflict related traits in our panel of hemigenomes. We measured two sexually dimorphic shared traits, namely locomotory activity and development time. And we also measured several proxies of persistence traits in males and resistance traits in females. First, we asked whether resistance traits are correlated with persistence traits. And unfortunately, regardless of how we measured persistence and resistance, we could not detect any significant genetic correlations between the two. For example, here you have male persistence expressed in terms of male mating latency on the y-axis. You have female resistance expressed in terms of mating latency on the x-axis. And clearly, there is no genetic correlation between the two. When we measured persistence and resistance in terms of remating latencies, again, we could not detect any significant genetic correlations between the two. And the story was somewhat similar when we investigated the genetic correlation between male mate harm and female mate harm resistance. So clearly, genetic correlations between persistence and resistance do not explain why we found a slight amelioration of intralocus sexual conflict upon strengthening the interlocus sexual conflict in the population. Next, we investigated whether resistance and persistence traits could have positive fitness consequences in the opposite sex. First, let's look at the consequences of male persistence on female fitness. In this graph, you have female fitness on the y-axis and male persistence expressed in terms of mating latency on the x-axis. In the case of the male bias sex ratio, which is the panel on the right, we could not detect a significant genetic correlation between mating latency and female fitness in the male bias sex ratio. However, in the female bias sex ratio, there appeared to be a significant negative genetic correlation between female fitness and male mating latency. Since lower male mating latencies are associated with higher male persistence, this translates to a positive genetic correlation between female fitness and male persistence. However, we think this is a slightly tricky result because on the one hand, you do have a positive genetic correlation between female fitness and male mating latency, but for some reason, when you go to an environment where there is intense interlocus sexual conflict, this correlation ceases to remain statistically significant. When we measured male persistence in terms of remating latency, we could not detect any genetic correlations between persistence and female fitness, irrespective of which sex ratio we were looking in. And the story was again somewhat similar when we measured male persistence in terms of male mate harm. Now let's look at the consequences of female resistance to male fitness. When we measured female resistance in terms of female mating latency, we could not detect any genetic correlations between male fitness and female mating latency, either in the female bias sex ratio or in the male bias sex ratio. When we measured female resistance in terms of remating latency, again, the picture was somewhat similar. However, things look slightly more interesting when we measured the correlation between male fitness and female resistance to mate harm. In the female bias sex ratio, we could not detect a genetic correlation between the two, but in the male bias sex ratio, female resistance to mate harm was appeared to be positively genetically correlated with male fitness. However, again, this correlation was not really statistically significant. So can positive consequences of resistance and persistence to fitness consequences in the opposite sex, to fitness of the opposite sex rather, explain why we found an amelioration of intra-locus sexual conflict upon strengthening the inter-locus sexual conflict? Perhaps, but we are not really all that certain. Lastly, we looked at whether changing the intensity of inter-locus sexual conflict resulted in a change in the selection gradients on traits that were shared between males and females. First, let's look at locomotory activity. Our results suggest that locomotory activity is indeed a sexually dimorphic trait, with males being more active than females. Very interestingly, there appears to be considerable genetic variation for the degree of sexual dimorphism for locomotory activity. In this figure, dots connected by solid lines represent a hemigenome line expressed either as female or as male. The panel on the left here is female bias sex ratio, and the panel on the right represents male bias sex ratio. So clearly, for a vast majority of hemigenome lines, they were more active as males than in females. However, there were a minority of lines for which the opposite seemed to be true, including an absolutely bizarre hemigenome line for which which was the most active as female and the least active as male in the male bias sex ratio. When we plotted this data slightly differently, to our surprise, we did not detect any significant genetic correlation between male activity and female activity. And we were very surprised to see this result because remember, locomotory activity has been previously shown to be involved in intra-locus sexual conflict in a population that was kind of related to the population we have been working on. Long and Rice had shown that locomotory activity was a shared trait in their population in the sense that there was a positive genetic correlation between male activity and female activity. But clearly, at least in our population, for some reason, activity does not appear to be a shared trait in the technical sense of the word, and therefore it can no longer mediate intra-locus sexual conflict in the population. Nevertheless, we still looked at the genetic correlations between activity and fitnesses in males and females, and there were a few surprises lurking here as well. For example, in the case of females, in the male bias sex ratio, we could not detect any genetic correlation between fitness and activity. But in the female bias sex ratio, there was a significant positive genetic correlation between female fitness and female activity. And again, this is an absolutely weird result because remember, our understanding so far suggests that less active females should enjoy higher fitness by attracting less male attention. In the case of males, however, we could not detect any significant genetic correlation between male fitness and male activity. And to summarize, locomotory activity clearly does not explain the pattern that we observed in our first experiment. Lastly, we looked at development time. And just like locomotory activity, development time too was a sexually dimorphic trait, with males eclosing later than females. And like locomotory activity, there appeared to be variation for degree of dimorphism for development time. For a vast majority of lines, they eclosed later as males than as females, but there were a few lines for which the reverse seemed to be true. Unlike locomotory activity, however, for the case of development time, we found a significant genetic correlation between male development time and female development time, suggesting that at least development time is a shared trait between the two sexes in the technical sense of the word. Our results from genetic correlations between fitness and development time also throw some interesting insights. For example, in the case of females, in the female bias sex ratio, we could not detect any genetic correlations between fitness and development time, but development time was negatively genetically associated with female fitness in the male bias sex ratio. But again, this trend was not really statistically significant. Males seem to mirror this result with no genetic correlation between development time and male fitness in the female bias sex ratio, but male development time was significantly negatively genetically correlated with male fitness in the male bias sex ratio. So taken together, these results suggest that as you move from the female bias sex ratio to the male bias sex ratio, the degree of sexually concordant selection perhaps increases in strength in the male bias sex ratio. And therefore, if all else remains equal, for which admittedly we do not have any evidence, then when you move from female bias sex ratio to male bias sex ratio, the strength of sexually concordant selection should increase, and therefore this would yield a lower signal of intra-locus sexual conflict. And therefore, in some sense, development time could potentially explain why we found an amelioration of intra-locus sexual conflict upon strengthening the intensity of inter-locus sexual conflict in the male bias sex ratio. To summarize, we found, to our surprise, a positive genetic correlation for adult fitness in all three sex ratios, which was a very surprising result because similar studies had previously shown that RMFs for adult fitness are either significantly negative or not significantly different from zero. Interestingly, we found a weaker signal of intra-locus sexual conflict at male bias sex ratios, in which the inter-locus sexual conflict was higher. We tried to investigate the mechanistic basis of this pattern, and we are not entirely sure if positive consequences of resistance and persistence traits to their respective opposite sexes, to the fitness in their respective opposite sexes, can explain this pattern. What we are certain of, however, is that correlations between persistence and resistance do not explain the weakening of intra-locus sexual conflict at male bias sex ratio, and neither does locomotor activity. And we think it is possible that the pattern we observed could potentially be explained by strong sexually concordant selection on development time in the male bias sex ratio relative to the female bias sex ratio. So a few people to thank. I would first like to thank my PhD supervisor, Professor N.G. Prasad, without whose unique brand of supervision, the lab would have been a very boring place. I would also like to thank all my colleagues from the evolutionary biology lab for their help during these experiments and for their feedback to improve the quality of this presentation in particular. I would also like to thank Professor Adam Chippendale for generously gifting us the clone generator system, without which none of these experiments would have been possible. ISER Mohali for giving us money to carry out research, and CSIR Government of India for giving me a PhD stipend. And thank you, all of you, for patiently listening to my talk, and I'll be happy to take questions from you. Thank you.
Wow, thank you very much, Manas. And that looks like a very healthy lab group you've got to work in there.
Yeah, everyone's not represented in this photo, actually, so we are even bigger.
Okay, so I've sent out the message to everyone to remind them that if they have a question, they can just relay that to me. But obviously, I have about a dozen questions, as well as maybe a quick comment. Comment is, first off, it's easy to be discouraged by the finding of a non-significant correlation in these kinds of data. You get a cloud of points and you go, oh my God, what does that mean? But it's important to remember that the null hypothesis is a one-to-one positive relationship between the sexes, where the same traits have the same impact upon female fitness and male fitness. And so that slope away, can be an indication that there is substantial intra-locus sexual conflict, but that there's also a mixture of intra-locus conflict, allelic variation, and positively correlated variation. Okay, so now the question. You've shown us a lot of correlations, and you've shown us some of these significant ones and some non-significant ones, but always what I'm wondering is, what are the heritabilities like within those data for female fitness, for male fitness, because a cloud of points otherwise could be, representative of error.
So we have not, at least I have not measured heritabilities for female fitness. I have not measured heritabilities per se for these traits, but we did measure heritabilities for female fitness and male fitness at 3 different sex ratios. I don't remember the exact figures, but there were considerably, we did detect non-significant heritabilities for all sex ratios for all sexes. As to whether there is indeed genetic variation for each of these traits, we actually fit linear mixed effects models for each of the traits. We did not explicitly measure heritability, but then in all the traits that I showed you today, there was a significant effect of hemigenome line identity. So in that sense, there does appear to be genetic variation for each of these traits. We had also measured mating rates in the sense that we had put a certain number of flies together in a vial, and we had tried to estimate mating rates using snapshot sampling, but that did not have a significant effect of line. So I did not show those results. But for the rest of the things, we did detect statistically significant effect of hemigenome line.
Yes, I would say that is an indicator that there is significant heritability in the fitness traits that you're measuring, which helps to shore up the confidence in the correlational data for me. So thank you for that. I see a question from Courtney Lanigan. Courtney, did you want me to read this or?
Oh, I can read it.
Oh, cool. Okay.
So to expand on your research, what related areas would you study in the future to continue with this project?
So right now, whatever experiments we've done so far, they are kind of single generation experiments in the sense that we have a large population sitting in our lab, and we are trying to investigate the genetic architecture for sexual conflict related traits in that lab, in that population. But our experiments so far don't really tell us anything about how these relationships are expected to evolve over several generations upon either increasing the intensity of inter-locus sexual conflict or reducing the intensity of intra-locus sexual conflict. So what I'm currently doing is to look at how signals of intra-locus sexual conflict evolve between populations that have been evolving for several generations, either under elevated levels of inter-locus sexual conflict or reduced levels of inter-locus sexual conflict. And these are populations that I actually mentioned some time ago, which Bodhi, a former PhD student from our lab, he has started these populations. And so basically he maintained one set of populations at a male bias sex ratio and another set at female bias sex ratio. And these populations have diverged over the last 270 odd generations. So I'm planning to look at the genetic architecture of those populations and see how signals of intra-locus sexual conflict evolve between these two kinds of populations.
Oh, very cool. Thank you.
Okay, I'm seeing Ian Chinsang with his hand up.
All right, this is a general comment, or maybe you can help me understand this. Is there really intra-locus sexual conflict? You gave examples in the fly, body size, locomotion, and timing. But if you use the term intra-locus, is it always coming down to one gene or one locus? Because there's always going to be interactions among that locus that you're studying. For example, body size. You can have one allele in the male, one allele in the female, and yes, the body size, but ultimately that allele might trigger a whole signaling pathway, and then it's not really intra-locus, unless I'm not understanding your term intra-locus sexual conflict. I know, we can think of it like this, because you mentioned if there is intra-locus sexual conflict, it eventually leads to sexual dimorphism. And we could say you could have the null allele, where, let's just use spermatogenesis, for example. A male might use a gene for spermatogenesis, and intuitively you would say the female doesn't need it. So you could delete that out of the female. And
yeah.
If the female doesn't really need it because it doesn't make those, but its fitness is now lowered, then do you still say, okay, you could say this is one gene, the gene for spermatogenesis, but the overall fitness is not, it's not reflected by this one gene. So that's the whole concept of is it somewhat of a misnomer to say there's intragenic or intragenic one gene itself, because ultimately it's an interaction of the whole genome.
Right. I mean, you are saying organisms are not just some of their genes. So, and what you observe at the organismal level is I don't know, an interactive sum of many different loci. So therefore, it's hard to predict what's happening at one locus from organismal level data. Is that what you're trying to say?
Yeah, and it comes down to what you're scoring too as your phenotype, because you gave the poster child of that water strider with the antenna. So the male is modifying its antenna to, grab the female's head. The female is modifying its head so it can escape that. But if we just look at the antenna gene, just that gene that controls that, that might have an effect on the female, its alleles, on how it's sensing. But then we would say, okay, if you just look at the antenna gene, you would say, oh, that's an intra-locus sexual conflict gene, just the antenna. But there's all those other aspects of that development.
Yeah, exactly. I think I agree with you in the sense that, and in some sense, that is what you were trying to look at. For example, if you think of intra-locus sexual conflict loci as just intra-locus sexual conflict loci, then perhaps you're losing out on some information. Or for example, as you said, the antenna gene typically is involved in inter-locus sexual conflict, but if it has consequences when expressed in females, you could potentially have an intra-locus sexual conflict between males and females. And now you have two different kinds of conflicts operating at the same time over one locus. So would you call it an inter-locus sexual conflict locus or an intra-locus sexual conflict locus? I guess that is exactly the kind of question that we attempted to address here. So I agree with you.
I mean, is there an example of, you gave those 3 examples in the fly, and 2 of them, it looks like you're disproving for the locomotion and the developmental timing, but is there like concrete examples of intra-locus sexual conflict?
I guess there have been not too many studies that have identified individual genes that are involved in intra-locus sexual conflict. I think there are a few. I don't know. Somebody else might be able to point out references, but I guess there have been a large number of studies that have looked for SNPs that are associated with antagonistic selection. And so that way there have been a large number of such studies, but identifying individual genes that are involved in intra-locus sexual conflict, I think, has proved slightly trickier.
Okay, thank you.
If I may interrupt, I believe there's a paper in Salmon, and the researchers found one single locus, one single gene that affects maturity time in males and females, oppositely in males and females, and it ties in directly to their reproductive output. It's Barton et al., 2015, if I may.
I think maybe too, Linda Dolph might know something about the genetic basis of the antagonism in that plant that Manas talked about, Silene latifolia, where they differ in floral size and leaf morphology. It's an absolutely brilliant example.
Yeah.
Okay, let's move on to a question coming from Tristan Long. Tristan, welcome.
I can't hear you.
You're muted.
Oh, yes. Still new to this. Hi, Manas. Really enjoyed the talk. I was wondering to what extent that you think the manifestation of inter and intra-sexual conflict are mediated by differences in physiological condition, and whether in your 3 different experimental lines, where the condition has changed in any fundamental way, or manifested, say, things like body size or things like that, or development time?
I am not entirely sure about physiological condition, but at least in our experiments, we did not have 3 distinct lines. We were actually using the same set of lines in different sex ratio environments. So in that sense, we did not really have samples from three distinct populations. We had samples from the same population, and then we were measuring, making measurements in those lines at 3 different sex ratios. But with respect to your question, I am not entirely sure. I mean, I don't know.
Chris Eckert has a great question coming up about the history of the lab populations deployed here. Christopher, do you want me to read it? Okay, Chris.
Oh, hi, Adam. Sorry, I was on the other side of the kitchen counter. I rushed over. Yeah, so thank you very much for that talk. That was terrific. You said that you sampled the 39 hemiclones from a single population. So I was wondering, was this a lab population that had experienced one environmental regime for a long time, and that would get rid of a lot of the kind of variation in fitness that you might find in nature? Or was it from a natural population where you might expect to see lots of variation for fitness, which would then generate those positive correlations you see between the sexes? Thank you.
So this this population traces back to a person called Larry Harshman, who I think founded it from 400 females or something like that. I'm not sure, but there are several replicates of this population, including in. Is there an LHM population in your lab, Professor Chipindale?
Yeah, we haven't had that population in the lab.
Okay, so
no. we did.
So there are. Oh,
Tristan does, yeah.
Okay, so there are several replicates of this population that are sitting in several different labs across the world. So yeah, they have been lab adapted for the last 20 odd generations, and for all these years they have been maintained in the same environment. But yeah, of course, it is entirely possible that the kind of genetic variation that is present in them now is not the same as what was sampled from those 400 females. But I guess there is considerable genetic variation in these populations, because whenever we have started selection regimes from them, you get a response. And even in this study, we did find significant additive genetic variation for fitness, and even for all these traits that we measured. So there is genetic variation present in the population, but is it the same as what you sampled all those years ago from the wild? Probably no.
Great, thank you very much.
Can I just add something there? I mean, this is one of those problems that has, possessed us and haunted us, all these years working on lab adapted populations. And you can argue it both ways. I mean, you could say that the lab adapted populations are really the ideal material for investigating a phenomenon like intra-locus sexual conflict, because you get rid of all of that stuff that's maintained by seasonal variability, by pathogens, et cetera. And you get down to, what's maintaining the variation that we have in the lab. And one possibility is this trade-off across the sexes, where you get good females, but they have low quality sons and vice versa. So it's sort of it's something that we're very interested in. Harsha is very interested in this question. It's an eternal question for us, and I know Tristan is too. But that population, LH, I did this work with rice way back in the late 90s. And at that time, that population was not maintained with any real careful regulation. It was just ups and downs in density and maintained all willy-nilly. And we found those negative correlations early on. Later, with the same population, we found that those negative correlations had disappeared. And that's a result that's been repeated in some derivatives of the LH population as well. So, maybe over extended periods of time, you've got a trade-off between the sexes, but ultimately, that allele that's just the best overall is the one that's going to win, even though it has a negative impact on the other sex. Okay, I thought Chris might answer back on that one, but.
So if I may add to that point, this is kind of weird, because if you look at the Connellon and Clark paper, it kind of argues in the opposite direction, in some sense, that they argue that as populations adapt to their environment, the degree of sexual antagonism should increase. But if you look at what is happening to these populations, then the degree of antagonism seems to be decreasing over time, over generations, at least with LHM kind of populations.
Yeah, again, that might just be that you've got these male benefit, female detriment alleles, and you've got these female benefit, male detriment alleles in populations. And over time, that may be maintaining a polymorphism for some period of time, but eventually the overall fittest allele fixes and you lose the variation. And then the hemiclone analysis is powerless to show us the sexual antagonism that's actually there. Oh, Maria, I see your hand is up.
Hi. So I have maybe a bit of an odd question, and this is me not really being too familiar with the topic, but you did mention that for intra and inter sexual conflict, there's good examples, and in some instances, specific genes have been identified. And I was curious if there's any common theme of the types of genes that can be involved in this type of conflict. Do they tend to be functioning in specific pathways or be specific types, like give rise to specific types of proteins, or is everything just strain, species, and sort of specific phenotype dependent?
I am not sure, but I guess all I can say right now is people have looked at tissue-specific gene expression and chromosomal distributions of sexually antagonistic genes, etc. But I don't know about specific pathways, but let's say, I guess people have identified SNPs associated with sexual antagonism for immunity in Drosophila melanogaster from Andy Clark's lab. And I guess they did identify a few of the genes located on the X chromosome of their flies. But other than that, I don't know if if somebody has investigated the patterns of what pathways these genes are involved in or what kind of proteins they code for. I mean, if there is any such study, I am not aware of it. I don't know.
Thank you.
Yeah, no, there have been, I think, some efforts in that direction, pulling things together. I could point you to some literature on that. A lot of what we've looked at, though, is sort of like the really crude kinds of genetics by your standards, Maria. That would mean, you know, like, is it the X or is it the autosomes? And burrowing into that a little bit more, you know, we had some data coming out of male limited evolution, a male limited evolution experiment where we kept the X chromosome and only selected for it in males. And some of the things that turned up there were strong associations with dosage compensation genes on the X. And there were some genomic regions, some chromosomal regions that certainly lit up. And we do have some candidates there. That's a paper we published last year with Jessica Abbott at Uppsala, sorry, Lund in Sweden. But yeah, I could definitely point you to those resources if you're interested.
Yeah, that's really interesting, because I was also wondering to some extent if a lot of the different phenotypes have been linked to SNPs to some extent, like they might not fall in genes at all, which then kind of gives into this idea of whether they're sort of like regulatory regions for something. Coming from a bit of an epigenetic background, like it could be places where modifications fall, or, you know, if they are genes, are they genes that tend to have more different types of isoforms, splice variants that can then give rise to like dimorphisms that are easier to see between males and females. I was just curious if any patterns were emerging yet at all for this type of work.
Early days, I think, for that on that front, but there's definitely a push to go there. I know in my lab, Harsha and I turned over generation 16 of a big male limited evolution experiment this morning. So we're hoping that selection will help to shed some light on what this particular genomic regions and actual genes and gene expression levels, you know, are associated with what we assume to be improving male fitness under the selection regime. So folks, I think the gong is sounding here for an hour. If we were really in session, that would have sounded 10 minutes ago at 50-minute mark, because people might be having to get to a class or something like that. So I would like to thank you so much for coming out today, Manas. Thank you so much to those of you that are attending late night from Mohali and other far-flung destinations. It's been a great seminar today. Certainly, you know, for the fly folks, but I think you did a superb job at laying out the background so that everybody could understand these two fascinating forms of conflict between the sexes. And I think that showed up too in the really excellent questions that you got at the end. Super job today. Really, really awesome seminar.
Thank you. Thank you very much. And thank you for hosting me. It was, I feel really privileged and lucky to be able to share my work with you. Thank you.
I find it very hard to end these things because, you know, what I'd like to do is to take you to the grad club over here or something and have a beverage of your selection. Do that. But I'd like to wish you a good night and thank you so much for putting this together.
Thank you.
Great.
Thank you.